Imagine placing a droplet of dye into water. At first, it holds together like its own tiny world. Then it dissolves outward, fading into the surrounding liquid. Now imagine something strange happens. The dye suddenly begins to coalesce again. It pulls itself back together, it changes shape, it dissolves and reforms repeatedly, almost like it's responding to its environment. If you saw behavior like that, you might instinctively think, is that drop alive? And in many ways, that's the essence of life itself. Not rigid structure, but dynamic organization. This is the core idea behind liquid-liquid phase separation. You can easily think of liquid-liquid phase separation as oil droplets in water. However, in biology, it's water droplets in water, and water droplets are filled with biological information. Inside living cells, proteins and RNA behave almost exactly like those droplets. They condense into liquid-like compartments called biomolecular condensates. These condensates form, dissolve, merge, separate, and reorganize continuously. They're not enclosed by membranes. They exist because molecules spontaneously organize into dynamic phases just like droplets in water. And this changes how we think about biology. Life may not primarily operate through static machinery, but through flowing adaptive matter. Nowhere is this more important than in the brain. Inside neurons, condensates regulate neurotransmission, memory formation, and synaptic plasticity. Your thoughts are not emerging from rigid circuits alone. They emerge from constantly shifting molecular landscapes. The brain behaves less like a fixed computer and more like a fluid information system. Now consider something even deeper. In wave systems like light and sound, information is distributed across patterns. Photons carry electromagnetic information. Phonons carry vibrational information. The information isn't stored in one location. It exists across the behavior of the entire system. Biomolecular condensates may operate in a similar way. Every condensate has a dynamic surface where molecules constantly interact and exchange information. Every droplet influences neighboring droplets. The entire cell becomes a continuously updating information network. Not static, not localized, but distributed across dynamic matter. This is why some scientists compare biology to a holographic system.